In the last segment, we will focus on another revolutionary topic, cancer immunotherapy. The 2018 Nobel Prize in Physiology and Medicine was awarded to James Allison and Tezuka Honjo for their discoveries of the inhibition of negative regulation of the immune system, leading to cancer immunotherapy. Let's take a minute to understand this concept. The immune system is our body's guardian, patrolling against bacteria and other foreign material that may invade it. The major players in this are T cells, who serve both as attacking cells and as regulators and coordinators of the immune response. T cells are also capable of recognizing tumor development in the body, but when a tumor does emerge, it often develops methods to suppress the immune system and overcomes it. By canceling this suppression, inhibition of the checkpoint on the T cells, the immune system cells can attack, destroy, and cure many tumors, even when they are metastatic. But what are the T cells attacking exactly? As you can see here, the center of this interaction between the T cell and the cancer cell is an antigen that is recognized by the T cells depicted in yellow. T cells are supposed to see foreign material to be activated. If they are activated against our own cells, this may lead to a condition called autoimmunity. Also, since cancer cells arise from our own cells, what are the foreign epitopes that lead to this recognition? The answer came from studying the attacking T cells in melanoma and other tumors, pioneered by Dr. Steve Rosenberg from the National Cancer Institute. Using biopsies of tumor and conducting both whole exome and transcriptome sequencing, we can find coding mutations that are unique to the tumor cells and hence produce unique peptides. We can also understand if and in what amount these peptides are actually expressed in. The peptides are now introduced into antigen-presenting cells and co-cultured with T cells that were isolated from either the blood or the tumor itself. These are tumor-infiltrating lymphocytes. Cells that reacted with certain mutations expressed activation markers, such as 4-1BB or OX40, and were further analyzed. This proved that T cells attacked peptides that originated from mutated genes. But most interestingly, most of these mutations were considered passenger mutations that are not oncogenic driver of the tumor, but considered non-significant and were shared by all tumor cells. Again, a change in a single base pair leading to a single amino acid change in a peptide is recognized by the T cells and leads to an immune response. These were very variable mutations with almost no two same mutations in different patients. Well, back to Dr. Allison and Honjo's checkpoint inhibitors. It appears that not all patients respond to these, but a certain subpopulation of patients in many tumors are extremely sensitive to this immune activating therapy. These are patients whose tumors bear a very high mutation load, here showing more than 10 mutations per megabase, with most more than 100 times compared to the regular tumors of the same type. With so many mutations, there is a much higher chance that at least one will generate a neoantigen that will wake up the immune system, and with checkpoint inhibitors, lead to cure. These are shown here in the gray circle. The number of mutations per megabase can be analyzed by genomic sequencing, and tumors with a high mutation load can be assessed this way. Here you can see survival curves of patients whose tumors were analyzed for tumor mutational burden, or TMB. Within a certain tumor type, patients with a higher mutation load, represented by the blue line, did better than patients with a low mutational load, represented by the red line. But there were tumors, as shown before, with an extremely high mutational load. The cause of these high mutational tumors is usually a defect in the mismatch repair mechanism, mutations in genes that are responsible for the repair if a mismatch occurs during DNA replication. Indeed, some tumors are able to impair this mechanism, termed mismatch repair deficient tumors. and these are the ones shown in the blue and black bars that respond best to checkpoint inhibition. This led to approval of these medications not for a certain anatomic disease, but for a cancer mechanism, mismatch repair deficient tumors. Today, mutational load, representing higher potential of neoantigen formation, and mutations in the mismatch repair genes are critical for clinicians to determine potential therapies for patients with cancer. In the practice session, you will get to examine the number of somatic mutations and to check the existence of mutations in the mismatch repair genes. In this unit, we were able to mention only a few aspects of the revolution that cancer research and treatment is undergoing due to advances in genomics and biomedical informatics. Clearly, we are far from victory in our war against this devastating set of diseases, but today we have much better tools to carry on the fight.